All right, so let's kind of explore some of these patterns of inheritance with human traits. So if you look in your lab manual, there's an appendix which gives you a whole list of these traits and how they're grouped. We're going to kind of talk about it. If you don't have your book, that's okay. What we would typically do is we would actually do crossing, and you'd pair up with someone, you'd produce some gametes, and you would see basically the genotype and the phenotype of your offspring. That's going to be a little difficult to do, but it's things I want to think about. So I just mentioned a couple quick vocabulary words. Let me just spell them out real quick. Then there's some of the things we're dealing with, concept of allele, right? So we hear often when we're talking about genetics and DNA and stuff like that, we hear about these base pairs and chromosomes and stuff like that. So how this all works, how it all organizes, you have those A, C, T's, and G's, right? Those are your nucleic acids. Those are the smallest unit of all that stuff. And there's these specific base pairing rules. And then if you have a whole bunch of those together, you get a gene. And a gene is a long sequence of these A, C, T's, and G's that codes for something. Right? It does something. It codes for something. Usually a protein, not always, but it does something. So we have a gene. So maybe you've heard that term, oh, you've got a gene for like blue eyes or brown hair, right? That's actually not necessarily correct. Typically, the gene is one specific product, and it may take many, many genes. It may only take one, but it may take many, many genes to actually influence a trait. And so that is where this term allele comes in. And we use allele to message or symbolize the variance of a trait. And it's a much simpler way. So you say, oh, you have the allele for that hair color. You have the allele for that blood type and things like that. It could be multiple genes. It could be one gene, but in general, it's just an easier way, a kind of a human-made idea of how to organize all this stuff. So an allele is a variant of a trait. And when we're talking about these genes and stuff like that, or when we're studying these genetics, there's two things you want to think about. We want to think of the genotype of the allele, and we want to think of the phenotype. Again, we're referring to an allele. The genotype is quite simply the code. Now, it's not those A, C's, T's, and G's that I just mentioned. This is an arbitrary code that's used to help convey messages. Something like a capital letter and maybe a little letter, or some combination like that. So the code itself. The phenotype is the actual physical trait. Right? So in other words, if we're talking about tongue rolling, this capital R, little r would be the genotype, but the physical trait would be the ability to roll your tongue. So can roll, let me actually write here, roll tongue. And so maybe right now you're sitting there trying to roll your tongue, and that's okay, roll your tongue into a tube. That is actually determined by your genes. There's muscle organization, neural coordination, stuff like that. But basically, the ability to roll your tongue into a tube like this, I'm doing it now, is dictated by your genes. And so the physical trait is the ability to roll your tongue. But the genotype is like this. And you can imagine, depending on the organization, you can actually have different genotypes for the same phenotype. So if we have big R, you can roll your tongue. If you have big R, little r, you can also roll your tongue. Okay, so two different genotypes give you the same phenotype and so on and so forth. Okay, so do remember those organizations, the arrangements. So now let's talk about these patterns of inheritance. And we'll start right there with rolling your tongue. So hopefully you finished practicing rolling your tongue. But let's just talk about some simple things. So another concept that comes up with genetics is these concepts of dominant and recessive. Well, so what does it mean? Well, so let's put up genotype here. Right? That's the code. And let's put up phenotype. So let's talk for the R's. So if you're looking in your appendix in this lab, there's a whole bunch of sets. Let's talk about this. So if you have a big R and then whatever else, your phenotype is can roll tongue. And again, that's like that. Putting it in a tube. I'm making funny faces. You can't see me. That's all right. Well, this is shorthand right here. So big R nothing is the same thing as big R or big R, little r. And both of these genotypes, you can roll the tongue. However, if you happen to have two of these little r's, your phenotype then is cannot roll the tongue. Well, let's pause here for a second. Why do we have two of these tiles anyways? What's going on here? Well, what these tiles represent, or these letters represent, and why we have two of them, because you get one from the sperm and one from the eggs. So each of the parents that contribute genetic material to the offspring, you get one from the sperm and one from the egg. And it's a random assortment. So if the parent has big R, little r, well, maybe in their sperm or egg, whatever, maybe they'll put the R in there. Or maybe they'll put the little r in there. And it all depends. There's lots of, it's mainly random. There's lots of stuff going on. But you can see that, depending on the genotype, will influence the offspring. And it influences the phenotype. And so that's why this matters, right? You can say, well, wait, who cares if you're big R, or big R, little r? For that person, you have the same phenotype that you can roll the tongue. But for your offspring, it does matter, because if you're big R, 100% of your offspring will also be able to roll their tongue. But only a percentage of your offspring, if you're big R, little r, will, depending on the genotype of the person contributing the other gamete, the sperm or the egg. Okay, so that's just stuff to think about. Now, this relationship right here is called dominant and recessive. Okay, so the big letter is dominant completely, complete dominance over the little letter. So that's why two big R's is the same as one big R. It doesn't matter. This is your standard, your classic dominant and recessive. Okay, so dominant, let me just put it so we can see this. So we have dominant and recessive. That's your standard dominant recessive. We just talked about that. So big R, whatever, and then little r. And if we want to be very specific, this is called homozygous recessive, because it's the same, homozygous. And then this would be, if you have big R, little r, this would be heterozygous. And you could also be big R, this would also be homozygous dominant. So on, okay? Just keeping in mind this dominant recessive. So here's a quick question for you to think about. Does dominant mean good? Does it mean common, like prevalent? Does it have any other meanings? What do you think of this dominant recessive thing? Well, let's just answer this by exploring the next one, Huntington's disease. So if you have a big H, whatever, remember here's our genotype up here. Here's our phenotype. If you have a big H and whatever, your phenotype is you have Huntington's disease. But if you have little h, no disease. Well, so right there with the dominant trait, you only need one copy of that dominant allele, and you have this disease. And what is this disease? Well, it's a autosomal, meaning it's not on the sex chromosomes. Dominant, meaning it's associated with the dominant allele. That leads to basically parts of your brain break down starting in your 40s and leads to paralysis and early death, loss of muscle coordination, things like that. It is not a good thing. There's no cure for it. And if you have it, you have it. And all it takes is one copy of this. Fortunately, it is very rare. Why does it still persist in populations? Well, So symptoms usually don't show up until the 40s, and maybe by that time you've already had children. And at that point, it might be too late. Nowadays, there are testing for it. I believe even some states require testing for it for marriage license and stuff like that. But 23andMe and those other type of testings will certainly test for it. But yeah, it is a dominant trait that results in a fatal disease that certainly can affect many, many people. Unfortunately, it is rather rare. So dominant and recessive only have to do with the relationship of the alleles and how they interact with each other and how they're expressed. Not good, not bad, or anything else. Okay. Now we're done with simple, dominant, and recessive. Now we've got some other things. Now we're going to talk about blood type. So think about this. How many types of blood are there? Well, there's A blood, there's B blood, and there's O blood. So how do we get these combinations if there's still, again, only two? You get two alleles, right? Well, you get more than two alleles. That's kind of a trick question. In this situation, we have these capital I, we're again representing a dominant, A. And in this situation, if you have another one of those, or you have a capital I, A, and here's the recessive, the I, little I, then your phenotype is type A blood. But we have these B's now. Big I B, or big I B, little I, you have B type blood. But to have the O blood, you have to have little I, little I. And so wait, what is this called? Oh, but we're forgetting another type of blood, aren't we? We're forgetting that AB blood. Well, how do you get this? Wait. So here's you have an IA, IB. And now, with these four types of blood, with these different combinations, now it becomes clear, since this can exist, since AB blood can exist, this is not your standard dominant recessive. And what this is called is, this is co-dominant. Because the big I whatever acts dominant, but it doesn't dominate over another big I. So big I A, big I B, they coexist. This is co-dominant. You can get a combination of two. The fact that AB blood does exist means it's a co-dominant. And what is this AB blood type stuff like that? Why does this matter? Well, these are like little flags that your blood cells hold on to mark them. And it's because there is things about this, right? There's specialties with these, and especially when you're talking about giving blood. Blood type AB is known as the universal acceptor. And blood type O is known as the universal donor. So how does that work? Well, what these things are is these are surface antigens. They're little glycoproteins. They're sugar proteins. Remember we talked about nutrition. There's these combination molecules that sit on the outside of your cell. They're like flags. They're markers that say, hey, I'm this type of cell. We talked a little bit about that with the CD4 positive cells with HIV. But that's what these are. These are these markers. And so basically, if you have a blood cell that's AB, or really let's simplify this first. Let's say we have blood type A. So here's your red blood cell. They look like little frisbees. They're like anucleus. But if you have type A blood, there's a little flag on there that says, hey, I'm type A blood. And so if you have an immune cell that comes along, it might recognize that A flag and be like, well, that's okay because I know we're type A. I'm used to seeing that A. We're all good. I'm out of here. Conversely, if you're type B blood, you have a flag that says type B. And your immune cells are used to that B type, so they see that. And again, hey, no worries, no problem. Now, if you're type O blood, you've got no flags. So your immune system doesn't see anything. It's like, yeah, whatever. I've got nothing to get worked up about. So what do you think AB looks like? Well, codominant, right? You've got both flags. You've got your A and your B. So what are those white blood cells used to seeing here? Well, they're used to seeing both A and B. And like, well, no worries, nothing here to get excited about. So because of that, remember, AB is that universal acceptor. Because why? What can happen? If I put A blood in there, well, there's just type A blood in there. And I don't get upset about that. I'm used to seeing A flags. Oh, if I put type B blood in there, oh, nothing to get excited about. I'm used to seeing those type B flags. What if I put O blood in there? No worries. I'm used to seeing A and B, so nothing to get me excited about. Everything is perfectly fine. That is why AB blood is the universal acceptor. But O blood is the universal donor because it's kind of the same idea. If the immune cell's looking for those A flags and they see no flag to get excited about, no worries, we're good. If the immune cells are looking for B flags, and again, nothing to get excited about, we're all good. O blood, however, can only receive O blood. So if you're blood type A, that means you can receive O and A. If you're blood type B, you can receive O and B. If you're AB, you can receive all. If you're O, only O. But hey, O is very common. I'm actually type O. It can go everywhere. And so that's kind of the reasons why, because your body has these immune kind of defense things. Whatever your blood cells are used to seeing, they need to see that stuff. They don't want to get excited by something they're not used to seeing. Because if they do, right, if you're A blood and you put in a B cell in there, your immune system's just going to lyse it. And it's no good. And that whole transfusion is bad, and it can cause actually all kinds of problems. If you look in your book, it also goes into the pluses, minuses. So maybe you know about negative and positive, like O negative, O positive. That's kind of an interesting side note. I'm not going to go into it too much since I'm making a video here. It's your standard dominant recessive. So if you have an RH plus dominant and you need 2 RH negatives to be blood type negative, it doesn't necessarily matter to you. But again, this is putting your blood in someone else. But where this gets really interesting is a situation where a mother who's developing a child, remember, so it's dominant recessive, so you only need one change. So let's say you have a parent, a mother, who is negative blood type. That means they have RH minus, RH minus. Well, if they happen to reproduce with someone and the sperm brings along an RH plus, that means the offspring is going to be RH plus because it has that one copy. Right? RH plus, whatever. Even though the mother's putting in the RH minus, the offspring is going to be RH plus. Well, is that big of a deal? Well, kind of maybe is. Because while the mother and the developing fetus don't share blood cells, white blood cells, those immune cells that we were just talking about, they actually do cross the placental barrier. And if they see this, well, they don't like it because they're used to seeing that. So this is no good. And actually the white blood cells of the mother can cross that barrier and attack the blood cells of the developing fetus. This can be a very dangerous situation. The first child, it's usually not that big of a deal. But once that immune system is exposed to it, now they're ready and they're primed to look for the RH plus. And that second child that also might be RH plus, well, there's going to be a very, very, very high response for the immune system, really aggressively attack the blood cells, and can lead to lots of health problems in the developing fetus. This is something that we can screen for. They can plan for it nowadays. There's medication that can suppress this, but this is a serious thing that happened for many, many years, all because of this incompatibility of blood between the mother and the developing fetus. Okay, that is incomplete dominance and blood stuff. There is a little thing in your book about tasting PTC. It's just kind of an interesting little side note that your flavor sensation is also genetically determined. Some people can taste certain chemicals. That's why some vegetables taste very bitter to them. That's why some people, if they taste cilantro, it tastes like soap and things like that. There's genetic configurations to how things taste to you, and it's very interesting. All right, so now let's talk about incomplete dominance. So we talked about codominance, we talked about complete dominance. Now let's talk about incomplete dominance. And incomplete dominance, the model that your book uses has to do with a certain condition. So if your genotype is beta A, well, your phenotype is no disease. If you're beta A, beta S, you have what's sickle cell trait. Oh, sorry. And your symptoms are very mild, if at all. But if you're beta S, you have the condition called sickle cell anemia. So I consider this, so this is incomplete dominance. Oh, sorry, my stomach is making noise. Incomplete dominance, and this is like a dosage thing. So the more copies of the beta S you have, the stronger the condition, right? This is like a mixture. It's not codominant where you get both. It's not complete dominance. This is like the more copies you have, the more of a dosage you get, or the stronger of reaction it is. And so, well, what is the sickle cell trait? What is the sickle cell anemia? These are proteins involved in blood cells again, and your red blood cells are misshapen to the point where they can look like little sickles. They can look like these little, like, half moon things. And so one thing is they don't hold oxygen as well, but another thing is they get stuck inside your capillaries, and it can cause basically anemia. Anemia means loss of blood oxygen, and it can just be total body exhaustion, high degrees of early premature death, loss of productivity in life. Your whole body feels exhausted all the time. It can be very, very severe if you have that full sickle cell anemia. However, if you only have sickle cell trait, the symptoms can be very, very mild, possibly none at all. In fact, there have been world-class athletes like NFL players, collegiate athletes, other things that have played professional sports with sickle cell trait. And in fact, it's so common, collegiate sports even screen for it. They do blood screening and things like that because sometimes you just don't even know you're a great athlete, but certain conditions can make things worse. Like there was a defensive end for the Pittsburgh Steelers many years ago who used to play, and he had sickle cell trait, and he was fine, an amazing athlete. However, his doctors would not let him go to Denver and play, and this was a big deal a few times because there'd be playoff games there. He actually missed a playoff game or multiple playoff games because he couldn't play in Denver. Why couldn't he play in Denver? The high altitude was less oxygen available. His blood cells were just not quite as good with the oxygen and didn't want to stress out his system, so he was not allowed to play. Well, so why does something like this exist? Well, we're going to talk about evolution in a next lab, but there's a very interesting give and take with this. It turns out that individuals with sickle cell trait have a higher resistance to malaria. And malaria is a vector-borne. It's carried in mosquitoes. It's a little blood protist. It looks like a little blob, kind of like the paramecians we saw in the microscopes on the unknowns for the microscope lab. These little protists, and they're parasitic. They go inside your blood cells and they destroy blood cells. They cause lots of problems. Still kills 500,000 to 1 million people every year. Still today, malaria kills hundreds of thousands of people. But if you happen to have that sickle cell trait, because of the proteins on your blood, malaria is less capable of getting into those red blood cells, and you are more resistant to it. So there's this whole give and take. And this is why you see individuals with sickle cell trait or sickle cell anemia more prevalent from places that have ancestry in parts of Africa and parts of Southern Asia where malaria rates are much higher. So you see this trait and the carriers for it much more common in those populations with ancestry in certain parts of the world. Why? Because malaria is still such a very, very, very dire condition. All right, the final thing we talk about for genetics, thank you guys for bearing with this long video, I erased genotype. The last thing to think about is the gamete production. So if you have XX, your phenotype is you produce eggs. If you are XY, and these are known as your sex chromosomes, you generally produce sperm. There's always exceptions in this biologically, but we're speaking biologically. If you happen to have a cell with two X's, that individual, the offspring, or that individual, the gamete type, should be producing eggs. If you have XY, you will produce sperm. Now, there's obviously variations of that, especially with other animals, stuff like that, but that's the general. These are the sex chromosomes. All right, so that's the basics of the notes for genetics. Thank you guys for bearing with this. A little bit of a longer video, but that's genetics.